What do these simple, hollow spheres have in common with living cells? Keep watching to find out. Hello, my name is Emma McDougal. I am a chemical engineering student at the University of Missouri-Columbia, graduating in May 2025. In summer 2023, I got to work in the Jang Lab as an RU student at the University of Florida, working with Dr. Youngsun Jang and Bonita Deb. Here's what I've been working on. These brightly colored hollow spheres are called vesicles. They don't look very much like the complicated diagrams of human cells you might remember from your biology textbook. But our goal in the Jang Lab is to disguise these simple vesicles by adding new features so they look like and behave like human cells. But why bother disguising these vesicles at all? Well, this project is part of a greater effort in science across academia, industry, and government agencies to design synthetic cells. Synthetic cells are exactly what they sound like, human-made materials that have the properties and abilities of natural cells. Synthetic cells are important because they can answer key questions in basic and applied research. They can help teach us more about the way natural cells and living systems operate. They can also serve as an adjustable platform to solve many real-life problems in medicine and biotechnology. Right now, these vesicles may not look or function much like natural cells do, but the Jang Lab team is working on adding new components to these basic vesicles that help them act like cells. Every piece of cell-like behavior that we can make these vesicles imitate adds to the disguise. Over time, when enough additions have been made to the vesicles, they'll be able to serve as synthetic cells and help us answer these key questions and solve critical problems. This summer, I've been working on one piece of the disguise for these vesicles, their ability to sense. Natural cells are able to sense and respond to their environment. I've been working on incorporating sensory functions into these vesicles and measuring the ability of the vesicles to sense. The new vesicles I've been making have a new sensory protein attached to them that hasn't been included in the basic vesicle formulation. We can make two types, red-colored vesicles with a sensory protein called FRB, and green-colored vesicles with a sensory protein called FKBP. Both of these sensory proteins can attach to a third component, an analyte called rapamycin. This small molecule has a binding domain for each of these proteins. That means a red vesicle and a green vesicle can snap onto each end of the rapamycin, creating a trimer, a group of 3 connected components. This is what a mixture of red and green vesicles looks like when imaged using fluorescent microscopy. And here's how those same vesicles look when we add varying concentrations of rapamycin. Visually, you can tell that the green and red vesicles cluster into groups and produce a yellow color when enough rapamycin is added. In the future, other scientists can use this work to build vesicles with different sensory proteins that can sense various small molecules that might be present in the body, such as insulin, glucose, or caffeine. By swapping out this sensory attachment coming off the vesicle, we can target different molecules. This work proves that it's possible to build vesicles that can respond to their environment by sensing small molecules. The next step in disguising these vesicles is to build in the ability to respond after associating with each other in order to release a drug or perform another measurable response. Every piece of the disguise that we add gets these vesicles closer and closer to becoming synthetic cells and unlocking a new area of science and engineering applications. I'm incredibly grateful for the opportunity to contribute to this work. I'd like to thank the members of the Jang Lab, the Department of Chemical Engineering at the University of Florida, and the National Science Foundation for their support. Thank you for spending time learning about my project.